Imagine being able to correct a disease, not with drugs or surgery, but by rewriting the very code of life itself. What once sounded like science fiction is rapidly becoming scientific reality. Thanks to the revolutionary technology called CRISPR-Cas9, scientists can now edit DNA with remarkable precision. In this video, we'll explore how this technology works, where it is being used, the benefits it offers, and the ethical challenges that come along with such extraordinary power. CRISPR has completely transformed the field of genetic engineering. For decades, scientists could only study genes and observe how they functioned. But CRISPR changed everything. Biology has shifted from simply understanding life to actually modifying it. One of its most remarkable features is its incredible precision. Scientists can now edit DNA down to individual base pairs, reducing unwanted mutations and improving accuracy. And perhaps most importantly, CRISPR offers hope for treating inherited diseases that were once considered incurable. Interestingly, CRISPR was not invented by humans. Nature invented it first. It originated in bacteria as a natural defense mechanism. These bacteria are constantly attacked by viruses called bacteriophages. To survive, they store fragments of viral DNA, creating a kind of genetic memory, and when the same virus attacks again, the bacteria recognize it and destroy it. Scientists discovered this system and transformed it into one of the most powerful gene editing tools ever developed. The CRISPR-Cas9 system consists of three major components. First, we have the guide RNA. Think of it as a GPS system that locates the exact DNA sequence scientists want to edit. Next comes Cas9. Cas9 acts like molecular scissors capable of cutting DNA with remarkable precision. And finally, we have the target DNA itself. Together, these components work in harmony to make precise gene editing possible. The process itself occurs in four main steps. First, the guide RNA searches through the genome until it finds the exact sequence it is looking for. Once the target is identified, Cas9 attaches itself to the DNA. Then comes the critical moment. Cas9 creates a precise cut in both strands of the DNA molecule. At this point, the cell's natural repair machinery takes over. Scientists can use this repair process to insert, remove, or replace genetic information, and it is this extraordinary ability that makes CRISPR one of the most revolutionary discoveries in biotechnology. One of the most exciting areas of CRISPR research is medicine. Scientists are investigating ways to treat sickle cell anemia by correcting mutations in the hemoglobin gene. Researchers are also exploring therapies for cystic fibrosis, a disease caused by defects in the CFTR gene. CRISPR has even entered cancer research, where immune cells are genetically modified to better recognize and attack tumor cells. These developments bring us closer to the possibility of permanent cures rather than temporary treatments. CRISPR is not limited to medicine. Its impact extends into agriculture as well. Scientists are developing crops that are resistant to diseases, pests, and environmental stress. Genes can be modified to improve drought tolerance, increase crop yield, and even enhance nutritional value. In a world facing climate change and population growth, these improvements could play a major role in ensuring food security. CRISPR has also become an indispensable tool in scientific research. Researchers use it to understand how genes function, to create disease models in laboratories, and to accelerate drug development. As a result, our understanding of genetics and molecular biology has expanded dramatically. Compared with older techniques, CRISPR offers several major advantages. It is highly precise, remarkably fast, and significantly less expensive. Its versatility allows scientists to use it in bacteria, plants, animals, and even human cells. These advantages have made CRISPR accessible to researchers around the world, accelerating scientific discovery at an unprecedented rate. However, with great power comes great responsibility. Although CRISPR offers tremendous benefits, it also raises important ethical concerns. One issue involves off-target effects, where unintended mutations may occur elsewhere in the genome. Another concern involves germline editing, which raises questions about so-called designer babies. There are also concerns about unequal access to these technologies, which could widen existing social inequalities. Therefore, scientific progress must always be accompanied by ethical responsibility. The future of CRISPR is incredibly promising. Scientists envision personalized medicine, where treatments are tailored to each patient's unique genetic profile. Researchers are also exploring new approaches such as epigenome editing, and beyond medicine, CRISPR may help create climate resilient crops and support sustainable ecosystems. But achieving these goals will require global cooperation and careful regulation. CRISPR-Cas9 stands among the most remarkable breakthroughs in the modern biotechnology. Its ability to edit genes with extraordinary precision offers hope for curing diseases, improving agriculture, and expanding scientific knowledge. But with such power comes an equally important responsibility. The future of gene editing will depend not only on what science can achieve, but also on how wisely human chooses to use it. And as we stand at the beginning of a new genetic era, one question remains. Will gene editing simply transform medicine, or will it redefine what it means to be human? Thank you.